Arrays. First, let's start with some definitions. A graph is a pictorial representation of interconnected objects. The objects are represented by points called vertices. The links that connect these vertices are known as edges. An array is a data structure that stores values of the same data type. Arrays are a popular choice amongst programming languages as they store data compactly and efficiently. An adjacency matrix is a square matrix used to represent a finite graph. It tells us what vertices are connected in our graph. Eulerian path. If you can walk through a path on a graph and use each edge only once, then that graph is said to have an Eulerian path. If the walk returns you to the starting vertex, it is called an Eulerian cycle. Graphs that possess Eulerian cycles are said to be Eulerian graphs. A graph contains an Eulerian cycle if every vertex has even degree. If two vertices have odd degree, it contains an Eulerian path, but not an Eulerian cycle. Hamiltonian path. If you can walk through a graph and reach every vertex only one time, the graph is said to have a Hamiltonian path. If the walk returns you to the starting vertex, it is called a Hamiltonian cycle. Finding a Hamiltonian path is difficult. However, checking if a path is Hamiltonian is relatively easy. All you have to do is follow the path vertex by vertex and check that it visits each vertex only one time. How does an array create a graph? Well, in the array above, each row represents one lettered node on the graph. To read the array, row A, position A, corresponds to AA. Row A, position B, corresponds to A, B, etc. Moving on to row B. Row B, position A, represents BA. If a given coordinate has a 0, that means that the letters in that coordinate do not have a line connecting them. In contrast, a coordinate with a 1 does have a line connecting them. By moving through the array, we are able to draw a complete graph. Looking in our example, row A, position A, has a 0, which means that A is not connected to itself. However, row A, position B, has a 1, which means A has a line connecting it to B, which we can see is true. Continuing to read the array produces a graph shown on the right. How do we code this? To represent a graph in code, you can use a 2D array. The ones and zeros are used in place of Boolean values, so a 0 means false and a 1 means true. Each line of an array represents a vertex in the graph. So in our example here, we have 5 vertices. The ones and zeros tell us what each vertex is connected to. It is the vertex's edge list. The vertices are also assigned a number corresponding to their position. So A is vertex 0 and E is vertex 4. In our example, we are asking the computer if it is true or false that node 1 and 4 are connected. These corresponds to vertex B and E. So the computer printed 1 to represent that it is true that these two vertices are connected. In contrast, the computer printed 0 to represent that it is false that nodes 0 and 3 are connected. Connection to psychology, mental disorders. An important feature of mental illness is that no two cases will present in exactly the same fashion. Of course, there are guidelines that determine whether an illness technically falls under a certain category, but nonetheless, there are infinite ways of expressing the illness. Likewise, an array has rules that govern whether it can be called an array or not. An example being that no node can connect it to itself in a simple 2D array. However, once an array has been established, the array and subsequent graphs can have endless combinations, especially as the number of nodes continue to grow. Connection to psychology, microarrays. Arrays are also used in DNA sequencing because they are used to examine the biological basis of neurological and psychiatric disorders. A microarray is an arrangement of biological samples that allow for thorough analysis of genetic information. Individual samples can be identified by their placement on the array, and the location of the gene is then recorded onto a computer database. Microarrays are useful in psychology and neuroscience because differences in gene expression between a normal brain and a diseased brain can be used to identify the molecular basis of the observed symptoms or traits. Microarrays are created by robotic machines that arrange thousands of small spots of DNA on a microscope slide. Each spot contains a unique DNA sequence that will recognize and stick to a different specific mRNA sequence. To compare the gene expression in two brains, one begins by collecting a sample of mRNAs from each brain. The mRNAs of one brain is labeled with a chemical tag that fluoresces green, and the mRNA of the other brain is labeled with a tag that fluoresces red. These samples are then applied to the microarray. Highly expressed genes will produce brightly fluorescent spots, and differences in the relative gene expression between the brains will be revealed by differences in the color of the fluorescence. The computer can relay all of this information back to us, and the data is used by neuroscientists to identify what genes differ between normal brains and diseased brains. This is beneficial because microarrays can store large amounts of information in an orderly manner.